Pifithrin-α. Pifithrin-α. An inhibitor of p53, TP53, cellular tumor antigen p53. p53 is a regulatory protein that is often mutated in human cancers. It prevents cancer formation, hence it is classified as a tumor suppressor. p53 is located in the nucleus of cells throughout the body, where it binds directly to DNA. When the DNA in a cell becomes damaged by agents such as toxic chemicals, radiation, or ultraviolet (UV) rays from sunlight, it determines whether the DNA will be repaired or the damaged cell will undergo apoptosis. In normal conditions, p53 is tightly regulated and maintained at low levels in the cell. However, in response to various stress signals such as DNA damage, oncogene activation, or hypoxia, its levels and activity can increase. Activated p53 induces cell cycle arrest, allowing time for DNA repair, or triggers apoptosis if the DNA damage is irreparable. This mechanism helps to maintain genomic stability and prevent the survival and propagation of cells with potentially harmful mutations. By stopping cells with mutated or damaged DNA from dividing, it helps prevent the development of tumors. p53 inhibitors target and inhibit the function of the p53. They disrupt the interaction between p53 and its negative regulator, mouse double-minute-2 homolog, MDM2, that binds to p53 and promotes its degradation, thereby regulating its levels in the cell. They prevent MDM2 from tagging p53 for degradation, leading to increased levels and activity of p53. Other examples of p53 inhibitors: roscovitin, celacyclib, induces apoptosis in many cancer cell lines at all phases of the cell cycle. It has been shown to down-regulate BCL2, MCL1, survivin, and XIAP and upregulate p53, p53AIP1, and BCLX etc. It has been shown to have a synergistic effect with other anti-cancer agents, such as doxorubicin, Taxol, 5-fluorouracil, cisplatin, and tamoxifen. NUTLIN3, an anti-cancer agent, selectively stimulates global DNA hydroxymethylation in TP53 wild-type cancer cells as manifested by the elevation of 5-hydroxymethylcytosine in genomic DNA. In contrast, it fails to enhance DNA hydroxymethylation in TP53 mutated cancer cells. Consistently, it only activates wild-type but not mutated TP53. Ellipticin, an alkaloid first extracted from trees of the species Ocrosia elliptica and Rauwolfia sandwicensis, which inhibits the enzyme topoisomerase E via intercalative binding to DNA. Cyclic pifithrin-alpha hydrobromide, a cyclic analog of pifithrin-alpha, sensitizes p53 deficient tumors to radiotherapy and chemotherapy, increases apoptosis in target cells when used in combination with antimicrotubule agents. Antimicrotubule agents block cell proliferation by acting on the polymerization dynamics of spindles of microtubules, which are important parts for many cellular functions including cell division. Pifithrin-mu also inhibits HSP70, with anti-tumor and neuroprotective activity.